All right. So again, thank you so much for joining us. All of you are on mute right now. You will stay on mute throughout just so that we give the speakers the opportunity to hear or to talk without some distraction. I would like to ask all of you to set your computers to the speaker view setting on Zoom. So if you go to the upper right-hand corner, you can see either gallery view or speaker view. If you click speaker view, that way you'll be able to see the actual video of the person who's speaking at that particular time. So take a moment to go into your upper right-hand corner and click speaker view. If you have any questions throughout this presentation, just go to the chat button down at the bottom of your screen and write in your questions. And hopefully once we go through all of our researchers, we'll have a chance to answer a few questions. So again, if you have any questions, just add them into the chat section. This will last probably about 45 minutes or so, I'm guessing, but we'll see how it goes since we've never really done anything like this before. So before we start, I would like to launch a quick poll just so that you have some context to why pediatric cancer research is so important to us. So all of you should see the poll right now. Of all the total government research funds devoted to cancer each year, how much is allocated towards pediatric cancer research? 2%, 4%, or 10%? So take a few seconds to vote. All right, so let me end the poll real quick. As you can see, most of you voted 4%, which is absolutely correct. So about 4 pennies of every dollar are devoted to, there we go, there are the results. Now 4 pennies of every dollar are devoted to pediatric cancer research every single year. So as you can imagine, that's not a lot. And that's really the void that we try to fill. And that's why pediatric cancer research and our Powered by Pablove Research Grants program is such a pivotal part of our mission. So with that, I would like to introduce the CEO of the Pablove Foundation, Kirsten Lynham, who's going to kick us off. Kirsten.
Hello, everyone, and welcome to our first ever Pablove Research Hour. I'm Kirsten Lynham, the CEO of Pablove, and I'm here to kick off our very special virtual event. For those of you who do not yet know me, I joined Pablove about a year ago after more than 20 years managing research projects and leading medical research institutes. I deeply care about research and I know a lot about the research process. So I say with a lot of confidence that the way we go about powering pediatric cancer research here at Pablove is special and important. Right now, this is an exciting opportunity for you as our community members and generous donors to get the chance to hear firsthand from our newest cancer-fighting researchers who are joining us here from the front lines to share with you the cutting-edge work that is coming from their labs. Just for some background, Powered by Pablove is our childhood cancer research grants program. Our model of granting is high risk, high reward. We invest in the most promising ideas from investigators with daring, novel ways to attack and solve pediatric cancers. They're often ideas that are so bold and so new that they won't receive traditional government funding. Through Powered by Pablove research funding, these enterprising researchers can further develop their ideas and become positioned to then successfully compete for additional funding. So how are we doing? Since 2010, Pablove has invested $3 million in cancer research funding, but that's not all. We are betting on winners. Our incredibly talented and impactful Powered by Pablove grantees have used our initial funding to bring in an additional $10+ million in funding. That's a net total funding of almost $13 million for curing cancer. We like to say that every $1 we invest today equals $4 in life-saving ideas tomorrow. We're about to start our research announcements, but before I will turn it over to our event host, Dr. Armenian, I want to mention that we have a surprise guest at the end of our Zoom. And let me tell you, you don't want to miss it. Dr. Armenian is a practicing pediatric oncologist, hematologist, and scientist at the City of Hope in Los Angeles. So he is our event host today, and he is also a board member of the Pablove Foundation, and he serves as the chair of our incredibly effective Pablove Scientific Advisory Committee. We are so honored to have him join us tonight for our new 2021 research reveal. And with that, Dr. Armenian, the floor is yours.
Is it okay? So I'm good to go. All right, perfect. Hello, everybody. Good afternoon, good evening, and for I think one or two people, very early good morning. So we have some international folks joining us as well. It's such a thrill to be a part of this session today. Thank you, Kirsten, Bart, Leah, and everybody else who has helped put this together. You're doing some remarkable things with Pablove, and I'm truly honored to be associated with Pablove. What I'd like to do is essentially set the stage for what we're going to be talking about today. My goal is to provide a brief overview and some introductions and really get out of the way and let the real stars of today speak about the research that they're working on currently, as well as what they're going to be working on as part of the sponsored work from Pablove. I do want to give you all just a background of how we select grants and how we vet the grant funding process, just so you get a sense about the behind the scenes work that has led to these awards today. Once a year, Pablove will send out a call out to the scientific community to send their best applications in. We initially have a letter of intent process, which basically essentially says in one page, summarize what you're proposing. Let's vet it to the first process. So we received 110 different applications as part of the first screening step. From then, we narrowed it down to 21 proposals that were invited for a full submission. Those 21 full proposals were reviewed by our incredibly talented scientific advisory committee, which consists of essentially who's who of childhood cancer research, not just in the US, but in the world. So some of the best minds in the country and beyond are actually vetting this research. So of the 21 applications that were reviewed, only the current 5 were selected. So less than 5% of submissions are actually made it through this stage. So you can just imagine that the folks we're going to be hearing from today are really, really stars and true top-notch researchers. And for those of you who have been supporting Pablove, just know that your funds and your support are going to be in the right hands. And our track record speaks for itself. I do want to say that, having been involved in the research field now for over 10, 15 years, it's really remarkable times that we're living in right now. Our research community is struggling due to a number of financial constraints and cutbacks that have happened both at the national NIH level as well as the foundation level. But it's so refreshing to see how Pablove not only has doubled down on research, again, doubled down on research, but has actually increased the funding and support this year. And in fact, this year is one of the most successful years we've had in terms of funding awards since the beginning of Pablove. So that's a testament to the hard work of the Pablove folks, especially Kirsten, who has really been a leader and a champion for research. So there will be no other organization, mark my words, who will be committing to research the way Pablove has in this year and for the future. So without further ado, I do want to turn it over to our researchers and our stars. So I'll be introducing them individually. They'll broadly sort of provide an overview of the work that they're doing. And then we're just going to go through them one by one and transition from there. And if you have specific questions about the work that they're doing, please keep that in your minds. We can, or type it in your comments section, and then we can get to it toward the end with all the panelists, okay? So without further ado, I'd like to introduce Dr. Vicky Huff. Dr. Huff is a professor in the Department of Genetics and a director of the MD Anderson's Advanced Technology Genomics Core in Houston. She has a remarkable research program. Her research focuses on understanding the genetic causes of human diseases, including the genetics of childhood cancers, such as Wilms tumor. So she'll be talking to us today a bit about the project that we have supported and some of the work that she's doing, focusing on the prevention of Wilms tumor in certain high-risk individuals. So without further ado, Dr. Huff.
Okay, thank you very much for the introduction. And I'm really happy to be here, and I am really excited about continuing the work that's now being supported by the Pablove Foundation. So as was mentioned, I'm a human geneticist in genetics at MD Anderson. And as also mentioned, my research program has primarily focused on Wilms tumor, which is a pediatric tumor of the kidney. And as part of that work, many years ago, about 10 years ago now, my lab developed a mouse model, the first mouse model for Wilms tumor. And we've used this model a lot to help us understand tumor development and the cellular etiology of the tumor. But now what we are proposing really for the first time is to use this mouse in a much more directed study that would be more impactful clinically, not just understanding tumor development, but as mentioned, we're wanting to see if we can actually prevent tumor development. So the mouse that we generated, the tumors in the mice recapitulate the alterations, the genetic alterations that are observed in tumors, many of the tumors in children with a very rare syndrome called WAGR syndrome. And these children are, they have several anomalies, but they are predisposed to Wilms tumor. About 50% of the kids will develop Wilms tumor. This is as opposed to about 1 in every 10,000 children in the general population. So these children, because of this very dramatic predisposition, they have to undergo screening by ultrasound, and for about 7 years, until they're about 7 or 8 years old, and they have to go in every 3-4 months. And it becomes very difficult for parents and the children because these children have other problems too. So anyway, we have this mouse model that really kind of replicates the tumors that these children get. And one of my clinical colleagues, Jeff Dome, which many of you probably know, had an idea of perhaps a compound that might actually prevent cancer development in these kids. But we really, before it could go and be used in a clinical trial, he felt like it needed some data from preclinical studies in mice. So that's basically what my Pablove Foundation project is to do. It's to carry out a preclinical study with our mouse model to see whether or not we can actually prevent tumors from occurring in the mice. And if the results are positive in this study, then it would pave the way to starting a preventative clinical trial in these WAGR children. I know I've interacted with parents groups, the International Wilms Tumor, the International WAGR Syndrome Association. People are very excited about this. And so I've been actually hoping that I could tell them that I'd gotten funding for this. There was a meeting last Saturday, but I couldn't until after this awards ceremony. But anyway, everybody will be very excited to know that this work in the mice is being funded. And I just greatly appreciate the support and the encouragement that you guys are providing. Thank you.
Thank you, Dr. Huff. Thank you for the summary. Really appreciate it. We're looking forward to the exciting results in this new paradigms of approaching high risk and cancer predisposition population. So we're going to turn our attention to Europe, actually. And I would like to introduce Dr. Armand, who comes to us actually from Barcelona, where it is nearly 3 o'clock in the morning. So we thought we were dedicated by staying up in the evenings, maybe staying after work a little bit more, but this is true dedication and compassion to Pablove and to the research. So Dr. Armand, thank you so much for joining us. She will be talking to us. She is she's a head of the department and attending in the pediatric neuroimmunology unit in Barcelona Children's Hospital. Her research is focused on identifying new diagnostic therapeutic approaches for nervous system disorders and inflammatory diseases. She's using new paradigms and new approaches to study very rare neurologic conditions that many people either don't have the know-how or the expertise to do it, and she really is uniquely poised. She will be talking about a grant that is funded through a special mechanism at Pablove through a collaboration with Crystal and Naveen Viswanatha, who I believe are on the seminar with us today. Their support of Pablove and their special resources that they've allocated towards a special type of funding is truly, truly remarkable. And so without further ado, I'd like to turn it over to Dr. Armand.
Thank you very much for this nice introduction. And I would like to thank the Pablove Foundation for the grant we have received, which is a very nice opportunity that will allow us to continue our research project in obsoclonus myoclonus syndrome. As you told us, I am Dr. Tyson Armand. I'm a neuropediatrician, and I work as a researcher at the neuroimmunology program at IDIVAPS Hospital Clinic in Barcelona. And I direct the pediatric neuroimmunology unit at Sant Joan de Déu Children's Hospital, University of Barcelona, Spain. As some of you know, obsoclonus myoclonus syndrome is a devastating neurological syndrome that affects people of all ages, but predominates in children with cancer, with neuroblastoma in particular. And this syndrome is an autoimmune, in this syndrome, an autoimmune response is highly suspected, but previous attempts to identify the antibodies or proteins involved in the autoimmune brain attack have failed to detect them, or if detect, they are only found in a minority of patients. We are a worldwide reference laboratory in identifying new antibodies involved in autoimmune encephalitis and paraneoplastic syndromes. And for this, in our study, we will use novel techniques approach using brain tissue and rodent and human cultures of neurons to identify antibodies in a group of 200 to adult and pediatric patients with obsoclonus myoclonus syndrome, whose clinical information and samples are already available. And we will also develop a murine model of passive transfer of a patient's antibodies and other molecules present in serum or CSF of these patients in order to reproduce the disease in mice. Such model will help to better understand the disease and to develop treatment strategies. And this is a summary of the project, and really we are very grateful to the Pablove Foundation for this grant, because it represents a very nice opportunity to continue our research in this devastating disease.
Thank you, Dr. Armand. I do want to put in a special plug by saying that there are very few scientists in the world who are as well trained and as innovative in studying these kinds of very rare conditions. And so we are especially proud to partner with you as you try to unlock the mysteries of some of these very debilitating and rare conditions. So we look forward to the results from your research. So thank you very much for joining us. Very much appreciate the late hours.
Thank you.
So I'll next turn our attention to Dr. Dongfang Li, who is an assistant professor and a CPRIT scholar at the Cancer Prevention Research Institute of Texas in the University of Texas Health Sciences Center at Houston. His research is really focusing on understanding childhood cancer biology using pluripotent stem cells, which he'll talk a bit about. He has some very innovative approaches on how we can actually understand how these stem cells actually transform into various types of cancers. The area that he will be focusing on is osteosarcoma, which is a bone tumor that we see in adolescents and young adults. And there have been very few advances that have been made in the treatment of osteosarcoma over the last several decades, despite our best efforts. So hopefully some of the work that he will be doing will help unlock some of the issues and the biology of this and help us treat this cancer better. So without further ado, Dr. Li.
Good evening. First of all, I would like to thank the Pablove Foundation Awards, the Childhood Cancer Research Award. I'm Dongfang Li, Assistant Professor and CPRIT Scholar in Cancer Research in Montgomery Medical School at the University of the Texas Health Science Center at Houston. Our research is dedicated to understand the cancer pathological mechanism by applying Li-Fraumeni syndrome patient-derived pluripotent stem cell IPS to model and dissect osteosarcoma, the most common bone cancer in the children and teenagers. Li-Fraumeni syndrome is a genetic disorder caused by germline mutation in the p53 tumor suppressor gene. Li-Fraumeni syndrome patients have the increased incidence to osteosarcoma, which provide a perfect disease model to study this malignancy. Also, somatic p53 mutations are common found in the clinical osteosarcoma specimen. Modeling human genetic disease have recently become feasible with IPS methodology, which was developed by Nobel laureate Dr. Shinya Yamanaka in 2006, characterized their ability to self-renewal differentiation to all cell lineage like embryonic stem cell. IPS provide a very powerful unlimited sources to generate differential cell and can be used to study disease mechanism, drug discovery, personal health care, and potential cell transplantation-based therapy. In the last 5 years, we have established the first human Li-Fraumeni syndrome disease model use a patient-derived IPS to define the potential pathological mechanism caused by mutant p53 in osteosarcoma. Our established Li-Fraumeni syndrome disease model provides excellent disease in the dish platform for study how mutant p53 can function as an oncogene to promote osteosarcoma development. So our main research project in the lab including the systems labels analyze and drug discovery of Li-Fraumeni syndrome and p53 mutant osteosarcoma. And also we are interested in system analysis. Mutant p53 drive the genome alteration during the osteosarcoma development. In addition to Li-Fraumeni syndrome, we also applied IPS to model other familiar cancer syndrome with osteosarcoma predisposition, such as retinoblastoma and Rosamond-Thompson syndrome. So in this current proposal, we continue to apply Li-Fraumeni syndrome and CRISPR-Cas9 genome edit IPS to study if the RNA methylation M6A is regulated by mutant p53. If the mutant p53 control the M6A play a role in osteosarcoma initiation and progression, we propose the model link M6A eraser ALKBH5 p53 mutation and osteosarcoma and plan to evaluate if the ALKBH5 inhibitor can be used to treat the Li-Fraumeni syndrome and p53 mutant osteosarcoma. Finally, I sincerely appreciate the Pablove Foundation organizing this wonderful virtual meeting, and thank you for your time.
Thank you very much, Dr. Li. Yeah, I think that there are incredibly novel approaches that you're proposing to study the osteosarcoma. And as you mentioned, I think the potential for what you're learning in the lab now and the application to other types of childhood cancers is especially exciting for all of us. So we look forward to how this will unravel and will continue to build on your already established successes. So we wish you much success in the coming year. We'll turn our attention to Dr. Natalie Wu, who is relatively early in her career, so we are especially happy to champion her work. She is an instructor at Children's Hospital Medical Center in Cincinnati, and her work is focusing on a relatively, again, understudied population of individuals. And as you can see, there's a theme here, right? Pablove invests in where the needs are the greatest, where diseases are understudied, where cancers are underappreciated, underevaluated, understudied. And so she's focusing on patients with neurofibromatosis, which is an inherited condition in about 1 in 3,000 individuals in the general population. What's among the many conditions that these individuals can develop is a relatively difficult to treat form of cancer called malignant peripheral nerve sheath tumor. And again, just like osteosarcoma, we've had very little advances in the cure for these types of cancers. But as she will tell you, the work that she's doing will hopefully help children, adolescents who are struggling with this condition live a better and healthier life. So without further ado, I'll turn it over to Dr. Wu.
My name is Natalie Wu, and I am a research associate instructor in the lab of Dr. Richard Liu at the Cincinnati Children's Hospital. So the disease that I study is called neurofibromatosis type 1. It's an inherited condition in children, affecting 1 in every 3,000 individuals worldwide. And one major burden of these patients are the growth of peripheral nerve tumors. Although these tumors are benign in nature, in some cases they can actually be transformed into malignant peripheral nerve sheath tumors, which would be a very difficult disease to treat that can kill 50% of the patients within 5 years. And patients with malignant peripheral nerve sheath tumors are very resistant to chemotherapy or radiation therapy, so making it a nearly impossible disease to cure. So, one of the promising ways to treat this malignant peripheral nerve sheath tumor is to target the specific signaling pathways that cause tumor formation and progression, and to discover potential drug targets against tumor growth. But we still don't understand a lot about this disease and how the benign tumors can be transformed into this malignant form tumors in children and adolescents. So in my proposal, I propose to use single-cell transcriptomics and epigenomics to enable us to detect global gene expression patterns of each individual cell and the gene regulatory networks that drive these benign tumor cells to become malignant tumor cells. So using single-cell approaches, we can actually follow the trend, follow the trajectory and the journey of a benign cell and how it becomes malignant by turning on gene regulatory networks. And I developed a mouse model in my previous work where I identify this signaling pathway called hippo signaling pathway. The downstream effector called TATSIAP is activated, and this actually leads to MPNST. And indeed, in human patients, MPNST has upregulated hippo signaling TATSIAP signature. So this mouse model, I can follow a low-grade benign tumor cell and how it becomes a malignant cell using single-cell analysis. And by combining with single-cell epigenomic analysis, we can also define the regulatory network that drive these cells to become malignant cells. And through this, we hope to identify targetable pathways to stop these cells from becoming malignant and to treat MPNST with targeted therapy. Another thing that I propose is also to understand why these malignant tumors are highly drug resistant. We know that in the benign form neurofibroma, using MEK inhibitor, it has been very promising, but MPNST is nearly not responsive to that. So I propose to understand the drug resistance mechanism using also this single cell analysis approach. And hopefully I'll be able to define how malignant transformation of this benign tumor into MPNST, what the mechanisms are responsible for that, and identify targetable pathways. And I'm really grateful for Pablove Foundation for supporting my research and helping me develop this project. Thank you.
Thank you very much, Dr. Wu, for joining us. We're looking forward to the exciting work that you're doing. It's truly ambitious work, and we are excited to see how this story unfolds, not just with the specific project, but beyond, because the potential is tremendous. So thank you very much. I really appreciate it. So our final researcher, Dr. Alexandra Stevens, was not able to join us due to a previous commitment. So you will just have to pretend that I am her. She looks nothing like me. So she definitely has long blonde hair is what I heard, which I am lacking. So put that aside for a minute, and I'll just tell you about her very briefly, about the work that she's doing, because it's just as remarkable as the previous four that we heard. So she is an assistant professor at the Baylor College of Medicine at Texas Children's Hospital, which is one of the best children's hospitals in the country. She is focusing on a specific cancer, childhood and adolescent cancer that we see is called acute myeloid leukemia, otherwise known as AML. This type of leukemia is very difficult to treat. Our overall cure rate is about 50%, so about half of children diagnosed with this cancer are cured long term. But even then, when you actually treat these children, you have to use very strong and very intensive chemotherapy drugs that in and of themselves can be quite lethal. About 5 to 10% of individuals will actually die from side effects of the treatment. So we clearly need to do not only a better job to improve our outcomes, but do a better job of how we actually treat them. She's actually looking at a novel out-of-the-box strategy. She's looking at there is an antibiotic that we use in pediatric cancer and adult cancers as well too, to help prevent a specific type of pneumonia. There's some reports that this antibiotic not only helps prevent a bad pneumonia from happening, but also has cross-reactivity that it can actually help treat cancers themselves, which is kind of an interesting finding in the lab setting, in mouse and other cell line settings. So what she's proposing to do is actually look to see whether or not this antibiotic, the use of this antibiotic, will improve outcomes in patients with AML. What's remarkable about this is that it's not chemotherapy, that it's kind of two birds with one stone. You not only prevent an infection, but also potentially improve the cure rates from AML. What's also special about this approach, aside from being sort of out of the box type of an approach, is that AML isn't just a cancer in children and adolescents, but it's also in cancer in adults. And it's actually one of the most common type of blood cancers that we see in much older individuals. So whatever we learn from this has the potential to translate to the much broader adult cancer community as well too. Again, another example of how the support and the work that Pablove is doing isn't just about the immediate sort of small community, but it's about the broader cancer community, and that's really, really remarkable. So I will end there, my end of it, but I would like to spend maybe the next few minutes or so just to moderate a session. For those of you who have questions for our wonderful speakers, please type them in. and then I will sort of read them out. There is one question that I saw from Lauren Bryant. It says it's for Dr. Li. And the question is it possible for your research to help us learn more about P53 mutations in brain tumor patients, not just osteosarcoma patients?
This is really a wonderful question, because one of my projects, we are working on the childhood brain tumor. So right now, based on our preliminary data, the same IPS disease model also can be applied to model the brain tumor. So currently we have the project also investigate how the mutant P53 is able to control the brain tumor development. And we also consider there is another mRNA methylation regulator called YDHDF2. It's quite different from the molecule I described before, LKBH5. So another molecule may play a role to regulate brain tumor development controlled by the mutant P53. Yes.
Thank you very much. Any other questions for any of our speakers? Okay, maybe I could just briefly I can ask. So Dr. Huff, can you tell me a little bit about what it has taken to get this idea off the ground, the idea of preventive research in this sort of understudied population? In terms of the community and the network that you have established, what's so unique about it? I mean,
well, yeah.
From what I remember reading your application, it's really remarkable that you were able to sort of, it's not just about, you know, your own lab, but it's really about a community of folks who are invested in this research.
Yes, it is. And in fact, the idea came about 2 years ago with this parents organization hosting a meeting. They hosted actually 2 meetings, and the first one was a larger meeting. It was mostly researchers talking about various aspects of this syndrome. The second meeting was a much smaller meeting, and it incorporated a lot of parents, and we sat around a large table and basically asked questions. The parents were asking questions to the clinicians, to the researchers, in terms of how they should be approaching the treatment of their children, things like that. And one of the recurring questions, and this was given to Jeff Dome because he's used to be the head of the clinical trials for Wilms tumor in North America, was this issue of how often should the children be screened and everything. And he said at this meeting, you know, well, actually, wouldn't it be nice if we could prevent this from happening in the first place instead of just screening for it? And that was sort of the catalyst for all of this. And they, one of the parent groups gave me some seed money to get started initially, to kind of get things going, with the idea that I would need to get additional funding to really complete the project. And so it's a really nice collaboration between clinicians, researchers like myself, and the parents groups. And to me, I find that very rewarding.
Yeah, it's incredibly refreshing, and I think that's a nice sort of way to wrap things up here. But and as a plea to those of you who are in the session, to who are not scientists but are sort of clearly interested, to know that it really does take a village and collaboration between the foundations and parent groups and sponsors and scientists, and it does take a village, and the numbers of you know, the funding that goes towards childhood cancer research is clearly not enough, and we need more engaged and motivated individuals to roll up their sleeves and really jump into this. It's incredibly rewarding to see all of you on the call, on this Zoom conference call. And we hope that you will remain engaged with Pablove, because as you can see, we are doing some amazing and remarkable things. And without further ado, I'll turn it over to Bart, and thank you all for your attention. Thank you for your commitment and your interest in the research that we do.
Great, thank you so much. Thank you so much, Dr. Armenian. Thank you so much, all of our researchers. It's really great to have you all join us this evening. Like I said, this is the first time we've ever done it, and it's really fun to put names to faces and names to projects. So thank you so much, or faces to names, I should say. So to wrap this up, I would like to introduce a very, very special speaker. I would like to turn it over to Yasmin, who is one of our Shutterbugs and a cancer survivor. So Yasmin, it's all yours.
Okay. Hi, everyone. Well, good evening, everyone. My name's Yasmin Layla. I'm 9 years old. I'm working really hard to become a singer, actress, model, and public speaker. I've been doing speeches for Pablove for almost 2 years, and I couldn't be more grateful for all they do for kids like me. I was diagnosed with acute lymphoblastic leukemia at 3 years old. I was in a bubble, as many of us are doing now with COVID. But it's simple for many of us just to wear a mask and wash your hands. Many will never know how it feels to go through cancer and how fast life changes for the patient, but also for the family too. During quarantine, it feels like I'm in a bubble again because we're at home and we're stuck inside. When I was going through cancer, I didn't go anywhere. The only people I got to see were my family. They were always supportive with me. It was a hard time, but my mommy always said, there's light at the end of the tunnel, even if you don't see it. I am a Pablove Shutterbug sweetheart. I joined this organization in 2017. I love taking pictures. It's like my go-to thing. My favorite views are form's eye, bird's eye, and my favorite is candid photos. It's when you take a picture without the person knowing. Shh, it's secretive. I am an ambassador for kids like me who are either still fighting for their life or who are cancer free. Thank you for all the doctors and donors who are keeping this organization going and keeping it strong. Just remember a few things for just to go on in life. Be happy, be bright, and be you. And always be kind, because you never know what fight someone's going through. That's it.
Woohoo! Wonderful, Yasmin. Thank you.
Everybody take yourself off mute and give her a round of applause. Good job.
Yay.
Well done.
Yay.
Yasmin, that was
Well,
absolutely
Yasmin.
awesome. You're such a champion on so many levels, and a
Thank you.
really tough act to follow. You're just gold. You are just gold, Yasmin.
Thank you. That means a lot.
But before we sign off today, I also want to extend our gratitude to all of you. You just heard about our impactful research, but I really want you to understand this can happen because you give to us. This is your harvest just as much as it is anybody else's. You support this work, and we couldn't be happier to have all of you in our community as well. Your giving hearts give hope for a cure. Just try that on for a minute. Your giving hearts give hope for a cure. We are very deeply grateful, and thank you again for spending this time with us. I wish all of you stay healthy, and as Yasmin said, wear a mask and wash your hands often. All right, thank you so much, everybody, and we will do something like this again. You all have a blessed evening, and all the good things may be with you. Bye-bye.
Awesome. Thank you so much.
Thank
So
you.
important. Thank you, Yasmin.
Thank you.
You rock, girlfriend.
You are awesome.
Thank you.
Thank you from Louisiana. Hey, Bart.
Thank you, everybody.
Thank you, everybody. Great seeing everybody's smiling faces.
Thank you, guys. Great seeing everybody.
Nice hair,
Naveen and Crystal.
I know, Naveen. Wow.
Yay.
Here.
Fantastic to have you. In Pablove gear, no less.
Oh, yeah. On brand. Great seeing everybody.
Good night, everybody.
Good night.
Take care.
See ya.
Bye.
Mm.
Bye.